Hello, my name is Matt Johnson, and I am an assistant professor in the Department of Biological Sciences at Texas Tech University, which is located in Western Texas. I want to thank the organizers of this symposium for inviting me, and I'm excited to share with you all our recent progress with Angiosperms353, a new tool for flowering plant systematics and biodiversity studies. The history of molecular phylogenetics in flowering plants is now about 30 years old and really took off with the widespread sequencing of RBCL. As a chloroplast marker, RBCL has been successful because it is relatively easy to isolate and amplify. It strikes the balance between primers that are stable enough to be universal while providing a good number of informative sites within most groups. Since that initial publication of RBCL phylogenies, there was a rapid expansion of available DNA sequence data for RBCL and other easy-to-amplify chloroplast genes like MATK. This graph from a few years ago shows the number of vascular plant species with one of those two markers to be in the tens of thousands. When sequencing RBCL and other plastid markers became widespread, it fueled the development of new subdisciplines. For example, the free availability of sequence data on GenBank led directly to supertree analyses, which built upon the data collected from narrower studies to build taxon-dense trees of all angiosperms for the first time. GenBank as a database also led to the development of meta-barcoding in plants, which has been used for identification of plants in environmental DNA and other species mixtures. Today, I think we sit at a similar position in plant biodiversity research. Genome-scale sequence data is becoming more widely available as cost of sequence continues to drop. These advancements put us in a position to reconsider many of our existing tools for phylogenetic systematics. For example, it's become well understood that there are problems inherent to single gene phylogenies like those constructed from RBCL or even whole organelle genomes. Deep coalescence occurs when gene copies from different species, shown here as the thin colored lines, coalesce differently than the species phylogeny, which is shown by the open bars. In the gene history on the left, the gene copy from species A in red coalesces with the gene copy from species D in blue, rather than coalescing with its sister species B in green. The other two trees show the coalescence of A, B, C, and D in different ways. And the problem is that when deep coalescence occurs, coalescent theory predicts that these three options occur equally likely. And that means that 2 out of every 3 gene trees may disagree with the species phylogeny. Luckily, there is a solution to increase the number of loci. But how do we do that? There have been a number of different methods discussed for increasing genomic sampling for phylogenetics. In plants, whole genome sequencing is still too expensive given the genome size of most plants, and as we discussed, standard DNA sequencing with amplicons is lab-intensive and may still not generate enough variable data. Reduced representation genome methods like transcriptomes and RADseq are useful for many studies, but may not be universally appropriate either because of high expense, reliance on fresh or living tissue, or producing an undesirable amount of missing data. Could target capture give the appropriate balance of cost and data to drive plant phylogenetics into the genomics age? If you're not familiar with targeted sequencing, here's a brief summary. First, we start with a shared genomic library. The DNA is broken into many fragments, each of which may or may not contain a gene of interest, represented here in blue. We design biotinylated RNA probe sequences that preferentially bind to the genes of interest. The hybrid DNA, RNA, biotin complex can then be physically isolated from the rest of the DNA library using magnetic beads. That means the sequenced DNA is now enriched for the genes of interest. Target capture has several benefits for biodiversity studies in plants. First, it is much more cost-effective than whole genome or transcriptome sequencing for most plant species. If you'd like to see how we arrived at this $30 US number, check out our recent paper in Apps here. Target capture sequencing can produce a large amount of data reliably. Typically, exon sequences are targeted, but non-coding regions are also recovered, expanding the phylogenetic scale for target capture. Finally, and very importantly for me as a herbarium director, target capture works great with degraded DNA like from herbarium specimens, because we're able to reassemble the small fragments from each gene after sequencing. You can read more about that in this recent paper from Brewer et al. We know that targeted sequencing works well across large phylogenetic breadths in plants. Many of you may be wondering how someone with the Twitter handle Moss Matters got involved with the development of tools for target enrichment in angiosperms. Well, I first worked on target capture in mosses, where we used probes targeting 150 nuclear genes to reconstruct an ordinal phylogeny of mosses seen here. One of the important discoveries we made in this paper is that even though the probes were designed from one part of the tree down here, we still got sequences from most of mosses representing 250 million years of evolution. When we directly compared our recovered sequences to available transcriptomes for data for some of the highly diverged species, we discovered that we can recover loci with target capture with up to 25% divergence. This gave us confidence that we could accomplish something similar with angiosperms. The first question was which genes to pick. We turned to the 1000 plant transcriptome, or 1KP project. They had been working on a phylogeny of all green plants using a set of 410 nuclear coding genes that appeared to be single copy in all of Virida plantae. Their phylogenetic analysis was impressive, so we knew that these genes would be useful for deep phylogenetics in angiosperms. However, the 1KP dataset had 610 angiosperms in it, and we couldn't design probes using every single species. Here's what we asked. If we chose species X, Y, and Z to be representative sequences in target capture probe design, would that be effective at recovering sequences from another species, like species Q? To help us choose these representative sequences, we used a k-meanoid clustering method to choose between 5 and 15 representative sequences for each gene. We were able to do this for 353 of the loci, which is how we got Angiosperms353. In our initial report, we demonstrated sequence recovery for 42 species representing a wide range of angiosperms. The probe sequences for Angiosperms353 are now freely available. You can see my GitHub and the Dryad repository associated with our paper. They're also available from Arbor Biosciences at a pretty big discount compared with custom probe designs of similar size. Full disclosure, I did receive a small collaborator discount from Arbor as part of the pilot testing phase of Angiosperms353, but I'm generally a fan of their products going back to the Moss Tree of Life days. In theory, the probes could be designed by any vendor. Speaking of our pilot testing, we expanded beyond the 42 species in the 2018 paper with the goal of representing every family of flowering plants with the Angiosperm353 genes. What you're seeing here is a heat map where the rows are samples and the columns are the 353 genes. The shading in each box is the proportion recovered of the coding region of that gene. The blank white lines represent some samples that we were unable to recover, mostly for reasons of poor DNA extraction. There wasn't much phylogenetic pattern to our poorly recovered samples, although because many of the Angiosperm353 genes have functions associated with photosynthesis, plants with fast-evolving photosynthesis genes, like mycoheterotrophs, were a bit tougher to recover. Our goal of assembling a family-complete phylogeny was not lost, however. This is where it's important to distinguish Angiosperms 353, the probes, from Angiosperms 353, the genes. We can still recover the genes from available whole genome and transcriptome data, as we see here for about 150 species with sequences on Sequence Read Archive, or SRA. When we add all of these together, we end up with 1,067 taxa, representing 415 of the 417 families recognized by the 2016 plant classifications in APG4. Our phylogeny, inferred using the summary coalescent method Astral, has high support for most familiar relationships within angiosperms, but also has a number of new arrangements and other regions where high support from plastid phylogenies are called into question. As I'm not much of an expert in angiosperm phylogenetic systematics, I won't linger on this tree too much. But if you'd like more information, I would recommend reaching out to Lisa Picorni, the lead author on this study, which is in prep. For the rest of this talk, I want to focus on where the plant phylogenetics community is with Angiosperms 353 and where we are headed. For example, while we have successfully extracted Angiosperm 353 loci from almost every angiosperm family, can target capture work across all 300,000 flowering plants? Are the loci variable enough to be used within narrower phylogenetic scales, like within genera or even populations? And should we be focused on these universal probes, or would custom designs for specific taxa be a better solution? This symposium is really timely because just last month we had the release of two special issues sponsored by the Botanical Society of America, the American Journal of Botany and Applications in Plant Sciences. Both invited papers on Angiosperms 353, and as one of the guest editors, I'm really pleased with the quality of work using Angiosperms 353 that is presented in these issues. Of course, I acknowledge that there are a number of great papers using Angiosperms 353 that came out a bit earlier or in other journals. However, I think the recent release of these special issues make it a great focal point for this talk. In the Angiosperms 353 AJB special issue, we clearly see a new wave of angiosperm phylogenetics with ordinal and tribe-level phylogenies for several different groups, including the orchids, common lineales, corneales, myrtales, and gentianales. I don't have time to go into all of these papers, but the gentianales paper by Antonelli, Clarkson, et al. Has a lot of the themes that can be found in the other papers. Within this order, there have been a long-standing conflict among earlier studies about the relative position of the four families listed here. The earlier studies all differed in which genomic compartment, plastid, mitochondrial, or nuclear, and the number of genes sequenced and taxon sampling. Using Angiosperms 353, the authors were able to more confidently resolve the relationships among these four families. Their result agreed with the 1kp analysis, which is not surprising since Angiosperms 353 takes its genes from the set used by 1kp. However, with the expanded taxon sampling enabled by target capture, we can see a bit more of the picture. Each of these pie charts represents the number of genes that agree or are concordant with the shown species tree. Here, where there is a major split representing the conflicting relationships from other studies, there is a large portion of this orange part of the pie chart representing discordant genes. This is exactly what we expect given the disagreement between earlier studies done with only a handful of genes. At the other end of the phylogenetic spectrum, several papers in the special issue focused on genus, species, and even population-level analysis. I'm of course biased, so I'll be focusing on the paper that came from my group on the potential of Angiosperms 353 for population genomic studies, which was led by Madeline Slimp, an undergraduate in my lab. In this study, Madeline sampled 95 specimens from 24 different species. All of the specimens are from a 50-year-old herbarium collection from the same region of Texas, the Guadalupe Mountains. Here you can see the heat map of gene recovery for each of the specimens, organized by species and by life history type. Recovery was weakest for one of our grass species that had tissue that was really tough to grind, so we think again that this is a result of poor DNA quality. Overall, the recovery of exon sequences in blue and flanking non-coding sequences in orange was really good and gives us confidence that there will be variation within species. Indeed, there was substantial variation within species for the Angiosperm353 genes, whether measured by pairwise nucleotide diversity, or pi, on the left, or heterozygosity across genes on the right. This really showcases the potential of Angiosperms353, as we were able to sample tissue from these 50-year-old herbarium specimens and learn something about their genetic diversity. We're excited about potential future use in conservation genetics of these rare species in the Guadalupe Mountains region. Next, I want to highlight a few studies that in the special issue which compared the utility of Angiosperm353 probes with other types of target capture designs. Taken together, these studies demonstrate that if you have a little bit of extra money to work on a specific group of interest, it can be useful to add orthologs of the Angiosperm353 genes for your target taxa in a custom design. This study by Hendricks et al. Was also interesting because they took two different sets of probes, Angiosperms353 and a more custom kit, and just pooled the targeted products and got the best of both worlds. Finally, I want to mention a few papers that show Angiosperms353 is following in the footsteps of RBCL by inspiring new analytical innovations. Here, a new method by Neuheimer et al. Called HybPhaser is able to identify alleles in hybrid taxa and phase the sequences. This is an important development because hybrid taxa are often excluded from phylogenetic analyses, especially when using many genes. HybPhaser can make sure that our studies of biodiversity don't exclude hybrids, even if target capture data is used. McClay et al. Developed some new tools to help with the sequence recovery of Angiosperms353 genes from target capture data. One of their tools, an expanded reference target file called Mega353, was able to recover substantially more loci than the default file, which they downloaded from my GitHub site. I was not upset by this at all. In fact, we used Mega353 in our own paper and found that it doubled the number of recovered loci for many samples. The incorporation of more and more sequence data to help with analysis is also highlighted by another recent publication, that of the Q Tree of Life Explorer. Many of the papers I mentioned were funded in part by the Plant and Fungal Tree of Life, or PAFTAL, initiative at the Royal Botanic Gardens, Kew. The PAFTAL folks have made all of the recovered sequence data freely available, along with information about Angiosperms353 genes, and even a tree of available sequences, over 3,000 species already sequenced after a few years. I think this truly highlights the collaborative nature of Angiosperms353, and that many of us are dedicated to seeing Angiosperms353 become the next major tool of biodiversity studies in flowering plants. Similarly, I'm excited that my own work with Angiosperms353 has led to some new collaborations that also parallel the development of RBCL and other earlier tools using identifying plants in DNA. One collaboration is with Dylan Schwilk and another professor at Texas Tech, and Akasha Feist at New Mexico State Las Cruces. Both Dylan and Akasha are ecologists interested in community assembly, and I'm working with them to determine if we can identify species in the soil seed bank by sequencing the soil or clean seeds rather than waiting for things to germinate out of the soil. Meanwhile, I've begun a collaboration with the US Food and Drug Administration, working mainly with Sarah Handy and Elizabeth Hunter, who are interested in identifying plants in mixed samples like herbal supplements and other over-the-counter products. In conclusion, I hope that I've shown that Angiosperms 353 is a powerful tool for flowering plant biodiversity studies. It's a cost-effective way to generate lots of sequence data, even from degraded DNA like herbarium specimens, and is useful at multiple phylogenetic scales within populations and across all angiosperms. Finally, I'm encouraged by the way in which Angiosperms 353 is inspiring new development of tools and new research questions, with the same spirit of shared data and code that helped earlier tools like RBCL become successful. And with that, I'd like to acknowledge and thank a large number of people, including the Q team with Bill Baker, Ilya Leach, Felix Forrest, and Paul Kersey, as well as Allison DeVault at Arbor Biosciences, who was great with technical support during the development of Angiosperms 353. The pilot phylogeny of 1,000 angiosperms is being led by Lisa Picorni, who is now at the Center for Bioinformatics and Genomics in Madrid, Stephen Dodsworth at University of Bedfordshire. Additional data analysis I presented here was done by Angela McDonnell and Norm Wickett at the Chicago Botanic Garden. And of course, I have to recognize Madeline Slimp, Haley Hale, and Lindsay Williams, who were lab members who worked with me on the population genetics paper I presented. Thanks once again for the invitation to speak in your symposium, and I'd be happy to take any questions.